All right. Hi, everyone. Good afternoon. I'm Abdul, and I'll be presenting on the tobacco mosaic virus and its impact on the tobacco plant. All right. So just an overview of what the tobacco plant is. So this plant belongs to the genus of herbaceous plants. They belong to the family Solanaceae, often called the nightshade family. These plants are indigenous to the Americas, Australia, Southwestern Africa, and South Pacific, but it's now cultivated globally in warm climates. So in regions where the climates are warm, these plants are able to thrive or grow. And just as I said, they are biologically perennial. So when I say they are perennial, they grow year round, right? So they are able to grow throughout the year continuously. But because of their commercial use, they are mainly produced for tobacco. It is grown as an annual crop. So after every year, the leaves are harvested to produce, you know, tobacco products like cigarettes and all that. Now, with the structure of the plant, it can reach heights of 1 to 3 meters, that is 3 to 10 feet tall. The stem is very thick and steady. So as you can see, the plant displayed there, it has a very thick stem and it's steady, right? And the entire plant is covered with sticky glandular hairs, which are usually called the trichomes. And this is a form of adaptation that helps the plant to fight or to defend itself against insect herbivores. So these glandular hairs are able to secrete chemicals like alkaloids that deter insect herbivores. And as you can see in the diagram, the leaves are very large, so they have very large leaves. They often exceed 50 centimeters in length, and they have different shapes. So they can come in different shapes like oval shape, they can be ovate, they can be elliptical and obovate. And looking at the arrangement of the leaves on the stem, it has an alternate arrangement of leaves, right? So you have the leaves arranged alternatively. And also it has an apical position when it comes to the flowers. So the flowers are positioned apically on top, on the apex of the plant. Now, as this is a plant pathology class, we also look at the chemical defense of the tobacco plant. So it has a very fascinating chemical defense system. It produces many phytochemicals that it uses to defend itself against, insect herbivores. And these include alkaloids such as nicotine, which is the main, you know, phytochemical it produces. Others include terpenoids. They also produce phenolic compounds and phytoalexins. Now, even though they have all these, phytochemicals that help them to defend against insect herbivores, there are other insects that have developed tolerance to these chemicals. So as you can see in the picture there, you have the picture of the Manduka sector, which is a caterpillar, and then also the picture of the aphids down there. These two insects have developed tolerance to the chemicals they produce. So for instance, for the Manduka sector, they have genes that are able to detoxify the nicotine that makes them specialists in feeding on the tobacco plant. And this also, from what I read, it helps them because they are able to sequester some of these nicotine. That makes them distasteful, and therefore it also helps to defend them against predators. So predators don't eat them or feed on them because they're able to sequester this nicotine to make them distasteful. Now, the tobacco plant is very important plant. So it's very important we talk about. Hello, Kamei? Dr. Musa, did you say something? All right, so the economic importance, or we have to look at the economic importance of this plant. So when you take the tobacco plant, its primary use is to make tobacco products, and these include cigarettes, cigars, pipe tobacco, and smokeless tobacco. And in 2024 alone, in the United States, projected to generate about 82.7 billion in revenue from cigarettes alone, and 9.71 billion in tobacco tax revenue. That is how important or how big tobacco is in the American economy. And when we look at the economic value of tobacco globally, it's valued at 904.9 billion in 2023 alone, and it's projected to grow. So there's a projection by 2030, the global tobacco market will be valued at 1.1 trillion US dollars. So it's a really big cash crop. Even though it has this high economic value, it also has some significant impact, and these include healthcare costs, loss of productivity, and then also environmental damage. Now we go to the virus itself. So the pathogen we are looking at here is the tobacco mosaic virus. And this virus infects a wide range of plants, especially the tobacco plant and other members of the Solanaceae family. These will include like the tomato plant and potato. They also belong to the Solanaceae family. So this tobacco mosaic virus is not only particular or specific to the tobacco plant. So it can infect other plants like the tomatoes or the potato. And when it infects plants, it produces these mosaic spots. So as you can see on the screen here, the leaves that are shown, labeled as B, you can see the spots there. It has these mosaic spots and this discoloration of the leaves. So it causes discoloration. So you can see how it looks brown instead of the green color. For some history of this virus, this is the first virus ever to be discovered and recognized when it comes to pathogens in plants, right? So it was first recognized virus disease of plants in the world and most common on the tobacco plant. And it's also the first virus where the protein coat, so the capsid, as you can see here in the structure, where the protein coat was fully sequenced into 158 amino acids. It was also the first plant RNA virus of which complete genome was sequenced. So it has its complete genome sequenced. So the entire sequence of the virus is known. Looking at it, what kind of plants does it infect? So like I said, the most common one are the Solanaceae family. So, but it infects more than 350 plant species within 9 different plant families of nutritional, economical, horticultural, and ornamental importance. And these will include weeds and members of the Solanaceae family, I mentioned earlier, these include the tobacco, tomato plant, we have the potato, the eggplant, the pepper, and petunia. Other families include the Brassicaceae, which include the cabbage and radish. We also have the Cucurbitaceae, which include the cucumber, watermelon, zucchini, and pumpkin, and also the legumes, leguminosae, that includes beans. Ornamental plants are also part of the plants that are infected by this virus. These include the chrysanthemums, then the impatiens. These are the two that are most susceptible to the tobacco mosaic virus. It's very important we look at the structure of this virus, right? So it's a rod-shaped virus. It has a rod-like appearance, and it's 300 nanometers long with a diameter of 80 nanometers. And it's covered by a protein shell called the capsid. Right? So when you look at this diagram here, this blue structure you see that is carrying the helical structure here is what we call the capsid. This strand that you see here is actually the virus's genetic material. And this capsid is covering it to protect it. So it's like a protective layer around the virus. And as you can see here, the genetic material here is single-stranded. So it has one strand, unlike, genetic material in animals that are double-stranded, or in other organisms that are double-stranded. This single-stranded RNA molecule. And the capsid is made up of 2,130 molecules of coat proteins that assemble. So they assemble in a rod-like helical structure that possesses 16.3 proteins per helix turn. So when you take each helix turn, they are made up of 16.3 proteins. And the RNA is found in a quelled manner. So as you can see here, it's quelled within the helix. So the RNA is found in a quelled manner inside the capsid coat and is made up of approximately 6,395 nucleotides. So this is just a picture showing you can see the RNA there, right, which is quelled, and then also what the capsule that protects the single-stranded RNA. So the mode of transmission, how is this virus transmitted into its plant, into the plant? First, the virus gets inside the plant host through vectors such as aphids, flies, and other insects. So insects that have these viruses in them, just like Dr. Musa said earlier in his lecture, some of these viruses are found within these insects, and as they feed, they introduce these viruses or pathogens in the plant. Transmissions can also be through susceptible weeds and plant reservoirs. So other plants that have these viruses can be agents of transmission. Apart from that, tobacco mosaic virus can be transmitted mechanically through the use of contaminated cutting tools. Also, if there are tools that are contaminated, it can be transmitted to a plant. Also, if you have maybe your hands are contaminated with the virus and you handle plant, it can be a mode of transmission. Plant cuttings, grafting, and contact between plants or other contaminated surfaces. Soil can also be contaminated by these viruses and can get into the plant, as well as potting material. So also look at the reproductive cycle of the tobacco mosaic virus, and it consists of 5 steps. These steps include entry into the host cell, uncoating, intracellular development, assembly or maturation, and release. So the first process is what the entry into the host cell. The tobacco mosaic virus cannot enter the host cell. It needs a mechanism. to get there. And there should be like a form of damage to the plants to allow entry, because we know plants have these physical barriers like cuticle on their surface that prevent direct entry. So there should be a form of break or damage to the plant cells to allow entry. And once this happens, once there is a breach in the cell wall, the virus has the opportunity to get into the cell. After that, it moves into the second process, and it's the process in which the capsid is removed and the nucleic acid is released into the cell cytoplasm. So when we looked at the structure, you see that it has a capsid in it, and the RNA, the genetic material, is found in there. So the capsid needs to be removed to allow the nucleic acid or RNA to get into the nucleus of the host plant. So once the capsid is removed, the nucleic acid of plant viruses enters the host cell cytoplasm along with the capsid. Now, when it's in the cytoplasm, the capsid is removed and the nucleic acid is released. And this nucleic acid actually gets into the nucleus. And to do that, it requires assistance using the host enzymes, right? So it's like the capsid needs to be degraded. And what it uses is the host enzyme, the enzymes of the host plant to degrade the capsid so that the nucleic acid or genetic material is released. Now, once this is done, it gets into the plant. Transmission of this, virus needs to move from one cell of the plant to another. To produce a disease, the virus must replicate. So it must, replicate or reproduce and spread to neighboring cells, right? Adjacent cells, and then they systematically move throughout the plant. Now, the spread of these virus to the neighboring cells occur through these channels right here. I think in our previous classes, we saw plasmodesma, and these are channels that link one cell to another. So these slender structures extending from the cell wall will connect adjacent cells, and this enables these viruses to move across from one cell of the plant to another. Apart from that, the TMV, tobacco mosaic virus, uses its movement proteins to spread from cell to cell, right, through the plasmodesmata, which connects plant cells and eventually enters the translocation system. So we saw the xylem and the phloem, right? So these virus get into the translocation system, and then from there it spreads into the entire plant. So it can get into the roots, the leaves, the stems through the translocation system. Then we look at how the viral genome replicates. So the tobacco mosaic virus contains a single-stranded positive sense RNA as its genome, right? And the replication of this viral RNA involves synthesis of a negative strand using the positive strand RNA as a template. So the original RNA strand found in the virus is a positive RNA strand, and it uses that positive RNA strand as a template to synthesize another strand, which is the negative RNA strand. And this involves two steps. The first step is synthesis of the negative strand. Like I mentioned earlier, it uses the positive strand as a template, and this forms a double-stranded intermediate termed as the replicative form. Then the second step is to synthesize a positive strand using that negative strand. So they synthesize a negative strand, then they use that negative strand to synthesize another positive strand. The RNA as a template using virus-coded RNA, and this forms the dependent RNA polymerase. So as you can see, this is a diagram showing how the replication occurs. So you have the positive strand here, and it is used to synthesize a negative strand. And that forms what we call the replicative form. Then it uses this negative RNA strand to synthesize a positive RNA strand. That forms the genomic RNA and then also the subgenomic RNA. This is translated to form the capsid proteins that protects the genomic RNA. After this, they need to be assembled. So after the positive strands RNA are synthesized, they need to be assembled. And this step is also known as the maturation step. So after intracellular synthesis of enough capsid proteins, so like we saw in the previous, they are going to synthesize proteins here, also synthesize genomic RNA. There is a need for these RNA that have been synthesized to be capsulated. So you synthesize enough capsid proteins, then viral RNA, and then the assembly process begins. So how is it assembled? They have to assemble the capsids around the viral RNA so that it is protected. And this process is highly organized. So initially, the capsid assembly begins when the capsomers associated with the 3' end of the RNA. So this is a diagram that is showing the process. Right? So you have the virus getting entry into it. Then there's the degradation of the capsule to release the viral RNA. The viral RNA gets into the host nucleus. Then there is synthesis of the negative strand. The negative strand is also used to synthesize another strand, which is the positive strand. Then there is translation. The translation leads to the production of the capsomeres that forms the capsid around the nucleus. Sorry, around the viral RNA. Now we look at some of the symptoms. What are some of the symptoms of this virus? So when a plant is attacked by this pathogen, what are some of the symptoms it shows? So it shows leaf necrosis. Sometimes the leaves curl, and there's malformations and stunted growth. And the typical, symptom is the mosaic patterns that are seen on the leaves, and then the loss of apical dominance. So as you can see, these are some pictorial presentations of some of the symptoms. You can see how the leaves are curling here, right? And then also the coloration. So that brings me to the end of the overview of this association between the tobacco mosaic virus and the tobacco plant. Then I'll present on the paper that I read on this. So this paper is titled Induction of Systemic Resistance to Tobacco Mosaic Virus in Tomato through Foliar Application of Bacillus amylopifaciens strain. That is the TBOG1 culture filtrate. So for this research, it's exploring a sustainable approach to protecting tomato plants. So one different thing here is this research was done on tomato plants instead of the tobacco. But one thing is they both belong to the same family. So that is one different thing in relation to like my first overview. Now, so for this, the researchers were exploring a sustainable approach to protect the tomato plants from tobacco mosaic virus using a beneficial bacterium. So the bacterium used there is a beneficial bacterium that can be gotten from the rhizosphere. And they used it to see if it's going to help the plant, the tomato plant, to develop acquired resistance against the tobacco mosaic virus. These plant viral infections pose a significant threat to agriculture. They reduce crop yields and affect security. And one of the most devastating plants it has effect on, I mentioned earlier, is the tobacco plant. There have been traditional control strategies such as breeding resistant plants and also using pesticides. But we all know these methods have certain limitations. So many chemical pesticides are ineffective against viruses, and they can also be harmful to the environment. So one promising alternative is induced resistance. So induced resistance against this virus. Like that is the plant, it's inducing the plant to use its own immune system or to activate the plant's immune system to combat against infections. So this study investigated whether a beneficial bacterium, which is the Bacillus amylocuficiens strain, can actually induce resistance against the tobacco mosaic virus in tomato plants. So for the hypothesis, the researchers hypothesized that the foliar application, so applying these bacteria on the surface of the plant, that is the leaves, would enhance tomato plant growth and even under viral infection. Two, reduce the tobacco mosaic accumulation in plant tissues. And then three, to induce systemic resistance through biochemical and molecular changes. To test this hypothesis, they designed a greenhouse experiment using the tomato plants and exposed them to the tobacco mosaic virus. And after that, they treated them with the T-bog1 strain of the Bacillus bacteria to see if it's going to help the plant or it's going to induce resistance. Now, for the objectives of this study, the primary goal of this study was to evaluate the effectiveness of the T-bog1 in inducing systemic resistance against the tobacco mosaic virus, and the specific objectives were assess the impact of T-bog1 on plant growth parameters, and these include dry weight, fresh weight, and then the root and shoot length. They also looked at to measure the disease severity, how severe the disease will be in the various treatment group, and also look at the tobacco mosaic virus accumulation in treated and untreated plants. They also looked to analyze oxidative stress markers and enzyme activities associated with plant defense, and also look to determine the expression levels of defense-related genes activated in response to the T-bog1 CF treatment. For the study design, the study was conducted under a controlled greenhouse condition, so the plants were grown in a greenhouse condition. And the plant used was the Carmen tomato cultivar, because this tomato species is highly susceptible to the tobacco mosaic virus. So it's not going to have any form of resistance against the virus. And the strain of the the tobacco mosaic virus used was the KH1. And this was mechanically inoculated onto the leaves to ensure uniform infection. Then the treatment that was used was the bacterial strain, that is the Bacillus amylocuficiens strain T-bog1, that was isolated from the tomato rhizosphere. So these bacteria are found in the rhizosphere of the tomato, and it was identified using 16S RNA sequencing. And the bacterial culture filtrate was extracted and applied to the tomato plant. So it was a foliar application. So for the experimental design, there were 5 treatment groups. As you can see here, we have the MOC R1, and these are plants that were inoculated with a buffer. So it's like a control group. No virus was inoculated in this plant and no treatment. So they were not treated with they were not inoculated with the virus. They were not treated with the T-box 1CF bacteria. Then we have the second treatment group, which is the TMV only. These are plants that were infected with the TMV, the tobacco mosaic virus, but they were not treated with the beneficial bacteria. Then we have the TB, that is the third group. This group were inoculated with the virus, and they were treated with the beneficial bacteria, which is the T1CF, before inoculation. So before the virus was inoculated, the beneficial bacteria was applied on them. Then for the fourth group, we have the TA. So these also had the the virus inoculated in them. So the virus was inoculated in this group, but the application of the bacteria, the beneficial bacteria, was after inoculation of virus. So TB was before inoculation, TA was after inoculation of the virus. Then we have the last treatment group, which is TBA. So for this treatment group, we have the TBOG1CF applied both before and after inoculation of the TMV virus. And as you can see, there were 5 replicates of each of these treatment groups. So we go to the results. So for the first table here, we are looking at the effects of foliar application of the bacteria on plant growth parameters. So for here, the plant growth parameters that were looked at were the fresh weight, the dry weight, the shoot length, and the root length. So as you can see here, for the control, comparing it with the TBA group, right? So the group that had the TBOG applied both before and after inoculation, you can see it had a higher fresh weight. That tells you that the plant was very healthy. So it had the highest compared to all other groups, right? And we can see the group, the TMV, the group that was inoculated with the tobacco mosaic virus, but wasn't treated with the beneficial bacteria, the TBOG1 culture. We can see it had the least fresh weight. And the trend continued when they compared the dry weight. The TBA group had the highest dry weight compared to the infected. Then when you compare the two other treatment groups, TB and TA, right, those that were treated before, so those that were given the beneficial bacteria before inoculation, showed a promising dry weight compared to treatment after inoculation. It was similar in shoot length, TBA having the highest compared to the one infected with the virus without any treatment. It had the least. Then the same applied to the root length. Right? So overall, we can see that the foliar application of the amylocufaciens bacteria, before and after inoculation showed a promising result. Then we also look at, the diseased effects of these strains. So as you can see in the mock, group that was treated with the buffer, you can see how healthy the plants look like. Now, for those that were treated with just the virus, so the virus was inoculated only without treatment using the beneficial bacteria. You can see how the leaves are, having this faded coloration, right, shows you that it is what diseased, right? Even though these two show some symptoms of disease, as you can see in the leaves here, it was better compared to the TMV group. That tells you that the beneficial bacteria is actually helping the plant to defend or be resistant against the virus. And you can see comparing all groups, you can see how healthy the TBA group looked, even though it was inoculated with the virus. Comparing it with the mock, you can see that it looks very healthy, because the beneficial was bacteria was applied to it before inoculation and also after inoculation. All right, this table is also looking at the disease severity, how severe the disease was, the reduction in disease severity, the relative expression of the TMV capsule proteins, and then also the reduction in TMV accumulation. As we can see here, for the disease severity, it was 0 here because these control were not inoculated with the virus. So there was no disease in these plants. So disease severity was 0. But comparing all groups that were inoculated with the virus, you can see that the disease severity was very less in the TBA. So when the bacteria was applied before and after inoculation, it decreased the severity of the disease. Also, we look at the reduction in disease severity, comparing the groups that were inoculated with the virus. You can see that the reduction was very high in the TBA group, 0% reduction in disease severity in the group that was inoculated with the virus without, foliar application of the bacteria. And then the expression level of the TMV capsule. So these proteins are produced by the virus, So if the expression level is high, it tells you that there is increased, number of viruses in the plant. So as you can see here, just 1 here for the control group. I don't know why it's 1, because this group didn't have the bacteria inoculated. But comparing the groups that were treated with the bacteria and inoculated with the virus, you can see that the TBA group had the least expression level of the capsid proteins, right? That tells you that it actually did really well. So the viruses were not able to replicate much in this group. But with the group that was not treated with the bacteria, we can see that it had 27, it had the highest, which is 27. That was very high compared to the rest. It was least in the TBA group. Then we also look at the percentage reduction in the TMV accumulation. Comparing the groups that were inoculated with the virus, you can see that the TBA group also did a better job. Right? So there was 90% reduction in the accumulation of the tobacco mosaic virus compared to the other groups. And it was 0% for the group that wasn't treated with the beneficial bacteria. Then we also look at this graph or chart. This was looking at the levels of oxidative stress markers. Here in this, they looked at 2 different oxidative stress markers. We have the malondialdehyde and the hydrogen peroxide. So these are stress markers. When a plant is stressed, they are going to produce more of these markers. So when we compare, we can see that it was less here in the mock group for MDA and hydrogen peroxide compared to all groups. Now, when we look at the TMV group, you can see that in both MDA and hydrogen peroxide, they had an increase or elevated, you know, stress markers. So increased stress markers for the MDA stress marker and the hydrogen peroxide in the TMV. And then when we compare, we can see for the TBA group, it was less, you know, for the treatment groups, the groups that were inoculated with the virus, it had what? The least, right? That tells you that the beneficial bacteria, help to reduce these oxidative stress markers in the plant. This figure also looked at, these enzymes. We have the polyphenol oxidase, PPO, we have the peroxidase, and then we have the SOD in tomato plants. And this was under different treatments at 21 days post-inoculation. And these enzymes play crucial roles in plant defense, and these help to neutralize the oxidative stress. So we expect it, if these are high in the plant, it means that, the plant's defense, is really strong, and it's helping to reduce oxidative stress in the plant. So when we compare the three, of course, it's going to be less in the first group, that is the mock group in blue. But when we compare the treatment groups that were inoculated with the virus, we can see that the TBA group had an increased or elevated levels of these enzymes. So the POX virus enzyme was elevated in the TBA group. It was the same for the PPO and SOD. And it was, compared, when we look at the PPO, it was least here for the TMV and then also for the POX. This graph or chart also looks at the impact of the TBOG1CF on the total soluble carbohydrate and total soluble proteins content in the tomato plant 21 days post-inoculation. And these, carbohydrates we know are important in plants because they use them for their metabolic activities, right? So they need these carbohydrates as well as proteins, the soluble proteins. So if they are elevated, it tells you that the plant is very healthy. So when we compare the groups, you can see in the TMV group, there was very least soluble carbohydrates compared to the TBA, which had the highest. Also, it was highest in the TBA group for the total soluble protein. Right? So had the highest for total soluble protein compared to all other groups. This tells you that the beneficial bacteria actually helped the plant. It's helping the plant in terms of growth and development. This graph also looks at ascorbic acid, and that is the vitamin C content, and the DPPH assay in the tomato plants 21 days post inoculation. And as we can see here, the high levels indicate oxidative stress, which damages the plant cells, right? So as we can see here, there is an elevated ascorbic acid in the mock group, right? It was least in the TMV, right? Then highest, when we look at the treatment groups that were inoculated with the virus, it was highest in the TBA group. These ascorbic acids help, the plant in terms of its defense mechanism. So this elevated ascorbic acid in the TBA group tells you that the beneficial, bacteria help the plant in terms of its defense. And it was the same for the DPPH. You can see there is elevated levels in the TBA compared to all other groups. All right, this chart also looks at the expression levels of key defense-related genes, 21 days post-inoculation. And these genes encode proteins which help fight against infections, right? So the first one were the pathogenesis-related genes, right? Just like I said, these genes encode for proteins that help to that helps to fight against infections. And the genes that were looked at were the PR1, PR2, and PR5. And when we look at the chart, we can see there is a significant boosted, PR gene expression in the TBA group compared to all other groups. You can see in the PR5, it was elevated. Apart from that, in the PR1, it was elevated. But I think for this, it was not elevated for the PR2 gene. And then it also looked at this other, defense-related proteins. These enzymes help to strengthen the plant cell walls and produce antimicrobial, compounds. Right? And now, as you can see, in the treatment groups, it was elevated in the TBA group compared to all other groups. So for the conclusion, based on the results they got, the Bacillus amylocufaciens TBOB1 induces systemic resistance against the tobacco mosaic virus. And then the TBA treatment group, so the groups that had the bacteria applied on them before and after inoculation, provides the strongest protection. You can also see that the TBOB1 CF enhances plant growth, it reduces oxidative stress, and activates the plant's defense genes. So for future research, they should explore the long-term stability of the TBOB1 induced resistance. So they have to look at how stable is it going to be? Is it going to have a long-term effect or induce a long-term resistance to the virus? And also field trials to confirm the commercial potential, because this was done in a greenhouse control, the number of plants were less. So if they can do a field trial to see if it has a commercial potential. But overall, the TBOB1 CF is a promising and eco-friendly alternative to chemical treatment for viral and disease management in crops. Thank you.
All right, thank you. So does anybody have any questions for Abdul? Obviously, it's a pretty good presentation. It had a lot of information. Probably one of the better studied plant pathogens. And here he's providing evidence that if the plant has this bacteria, provides more resistance to a virus than some of the mechanisms behind that, or correlations anyway. So who would like to ask a question or add some insight? This looks like Edit might.
Okay, Abdul, nice presentation there. So,
Thank you.
All right. So I was looking at the TBOG board, right? So
Yeah.
that induces this resistance. All right. So does this effect, can it be transferred to the next generation, like to the, if you're to pick a seed from this plant and then you plant it, will the planted seed have the same resistance, or induced, yeah, resistance, or it only ends with the parent plant.
Okay, so if I got your question right, if would this resistance be transferred like to future generations of the plant?
Yeah, yes, yes, exactly.
I'm not so sure about that, but I think it should be possible that this form of resistance is transferred to the next generation. I don't know if the resistance is like on a genetic level, but as you can see, they looked at various resistance genes and different kind of genes, and this is like more of a molecular level. So it's possible the resistance can be transferred to the next generation.
All right, thank you very much.
All right, who else has? Looks like Michael has one, and so does Elena. So, who would like to start?
I have a quick question about the genetics behind the virus. Viruses are known to their genomes change frequently and quickly. So one question would be, have you found the genome for the tobacco virus to change very quickly, number one, and number two, if it does, how much work is it to stay on top of those changes given the genetic component for your research? Thank you.
So your question was if because viruses are known to like undergo mutations, does the plant also have able to let's say co-evolve in response to these mutations? So I'm not sure. This is not actually my work. This is like someone's work. So I'm not so sure. That is why in the limitations I was like trying to see if they can do it commercially to see, because if this was done in a controlled environment, just for a short period of time, and most of these parameters they took was just 21 days after inoculation. Right, so it's possible 21 days might be a shorter time for the virus to mutate and develop a resistance against this systemic defense. So I'm not so sure, but I think that would be like a future research they'll have to do, and maybe modify the experimental design so that maybe they can take this data after a long period of time, not just 21 days, so that if there is any form of mutation, the effect can be seen, right? And then you ask the second question.
Oh, and how much work would it be to stay on top of those changes? Actually, I think you probably answered the second question too. If you had enough data over a long enough period of time, you could somewhat predict what the changes might be, and that would help
Yeah.
along the line too. I think you kind of answered it, but thank you.
Okay, thank you.
So I have a question. So let's say humans were to harvest tobacco that has the virus on it, and they were to like process it into like cigarettes and stuff. Would that have an effect on humans if we were to inhale it? And would it have an effect on non-infected tobacco plants if you were to smoke next to if you were to smoke a cigarette that had the virus, if you were to smoke it next to a tobacco plant, could that affect it as well?
Okay, not a great question. So one thing I know when I was going through this research was the virus is strictly a plant pathogen, so it infects plants only. So I don't think it should have like any effect on or any form of pathogenicity on humans. But with your second question, one thing I know for sure is the virus is a very stable virus. So it depends on what conditions. So for instance, if it's dried tobacco leaves, it can remain persistent. We know viruses with this capsid they have can remain persistent until like they get into a host, right? So viruses outside the host is like a particle. They behave non-living, right? But when they get the opportunity to get into a host plant, they begin to use the plant mechanisms, to reproduce or replicate. So it depends on the condition. If it's like, let's say I'm smoking tobacco and some of these leaves just drop down, right? It's possible if it contains the virus and gets into a host plant, they can thrive. It can affect a new plant.
Yeah, when we were working with tobacco, if you had somebody that was a smoker, there's a potential that they would have the tobacco mosaic virus still on their fingers from a cigarette and could spread it. So it's actually very possible you'd have to have extra precautions for smokers working with your research because they could spread the virus unknowingly.
Okay, thank you.
Who else has a question for Abdul before we move to our next presentation? All right. So, Elena, do you want to give it another shot? Or
Yeah, I'm just gonna look at the notes on my phone, but I will make it look as natural as possible on
Sure.
the presentation. So
all right. So we'll end the recording for this, and then so Abdul, end the recording and please make Elena the host.
How do I do that?
Go down to the bottom first and stop the recording.
Okay.